And the way I'm trying to look at the linkages of microbial physiology to ecosystem processes is using a trait-based framework. There is a trait-based framework in plant ecology, which was put forth by Grime in the 70s. And this is the classical CSR framework, the competitor ruderal stress tolerator framework, in which they really put forth how along two environmental gradients of stress and disturbance, plant species can structure themselves along these three main functional groups. And these are competitors occupying the space where stress is minimum and disturbance is less. So they can grow really well. They outcompete other species. In environments where the disturbance is very high, but stress is low, ruderal species such as grasses, which have a high turnover, can really persist well and occupy that niche space. And in environments where disturbance is low, but stress is chronic, stress tolerators are able to persist and thrive. And they also suggest that that in places where disturbance is really high and stress is also chronic, plants don't really survive. And there are many spaces like these on the planet. So a similar similar trait-based framework would be ideal for microbial ecologists as well, to study how microbial species structure across environmental gradients. But as much as we want to impose or map the CSR framework on microbes, we also realize that there are some inherent problems, because microbes are, or at least soil microorganisms, are mostly heterotrophic, whereas the CSR framework applies well to autotrophs relying on solar energy as a main source of energy. For microbes, there are a different sort of traits that might be very important. Microbes rely on an external source of carbon and energy, so resource acquisition is a very important trait. Growth yield can be a trait which is very similar to the competitor trait, but the competitor trait doesn't map well on microorganisms because there can be fast-growing microbial species which are not necessarily high yield, in the sense that they're not very efficient. Their growth can be very leaky. And stress tolerance is one of the traits which can be mapped well from the CSR. This is because microbes experience abiotic stress in the same way as plants do. So the abiotic stress that microbes express would be seen as a stress tolerance trait, or a life history strategy that encompasses some of the traits for stress tolerance or stress avoidance. So keeping this perspective, we put forth a framework where we chose these three life history strategies along two main environmental gradients. So this is the YAS framework, the high growth yield, resource acquisition, stress tolerance framework to infer life history strategies of microbial populations along environmental gradients. And the two axes that we chose were stress and resources. Resource because microbes, like I said, rely on an external source for carbon and energy. And what we predict is that in places where stress is minimum and resources are ample, the high growth yield strategy will excel. In places where stress is high, but resources are limited, resources are ample, stress tolerance or stress avoidance life history strategy will prevail. And in environments where stress is limited, but resources are ample, or resources are limited and stress is minimum, stress resource acquisition traits will dominate, and hence the resource acquirer life history strategy will be more dominant. And in places where microbes experience high stress and less resources, microbes can still persist, and that's the beauty about microbial physiology, that they are everywhere. So there will be a combination of the S and A strategy for that. And the basis for this is that there is a trade-off. This is the cellular level allocation of resources. Under limiting resource conditions, microbes will have to decide where they invest their resources. So consider a microbe, a cell that's lying in the soil, surrounded by some nice polymeric substrates. They break it down using extracellular enzymes into monomers or oligomers, which are then taken up by the cells using transporters. And then microbes use it for assimilation or dissimilation. And once precursor compounds are formed, they can decide whether to use it for biosynthesis and reproduction, or whether to use it for non-growth production, which is mostly maintenance purposes, such as extracellular enzyme production or investment into a lot of stress tolerance factors. So we hypothesize that there is going to be a trade-off at this stage, where the microbe really makes a choice where to put their resources. And like I said, in various environments where there is more stress or limited resources, there will be a trade-off against growth, and therefore the growth yield will be lower, which then has consequences for ecosystem level processes such as decomposition or soil carbon accumulation. So now I'm going to show two examples of how we applied this trait-based framework. First, in the context of UK, this is a nice study which was part of a bigger project which was funded by NERC Soil Security Program, and I became part of this bigger project and did some nice analyses of microbial traits along land use gradients. So what we did here is sampled across the UK 28 sites, and each site had a paired contrast. And this contrast was mostly local with a fence, contrast of land use intensity. In most cases, it was a more pristine environment next to a highly intensive arable grassland. And what we see here is that land use intensification can lead to a loss of carbon because of tillage and other very intensive practices. So we really wanted to see if the microbial ecophysiology in these differing systems is different, to really figure out the impact of land use intensification on microbial metabolism and how that relates to soil carbon cycling. The beauty of this use of local contrast is that it allowed us to really isolate the impact of land use intensification while the climatic conditions and the parent material remains the same. So what we did here was measure the community carbon use efficiency. Triplicate soil cores were extracted from the soils, and this was an incubation experiment done in the lab. We added 13C labeled plant litter, which was converted into a solution of plant litter DOC. And this litter was then added to small microcosms. And then we measure the amount of 13C that goes into the microbial DNA and the 13C that's in the respired CO2. And that allowed us to measure the community carbon use efficiency, which is a proxy for microbial carbon use efficiency. And we also used it to measure turnover rates. And what we saw here was that if we try to relate the carbon use efficiency to soil carbon, we found that there was a big cloud. But once we separated, once we looked at the different parameters that were actually determining the distribution of carbon use efficiency, we found that soil pH was a very important factor, and that was distributing the carbon use efficiency data into two data sets, one which was high carbon use efficiency and one really low carbon use efficiency. And there was a threshold at around pH 6.2. So what we saw is that below pH 6.2, there was no covariation of carbon use efficiency with soil carbon, but above pH 6.2, carbon use efficiency nicely correlated with soil carbon. And so we hypothesized that there are two different mechanisms of soil carbon storage across this pH threshold. Below pH 6.2, there is mostly retardation of organic matter decomposition because there is minimal microbial growth. And above pH 6.2, because there is a nice correlation of microbial carbon use efficiency with soil carbon, we suggest that higher growth efficiency, higher growth yield leads to increased microbial biomass, and that then leads to increased soil organic carbon accumulation in such soils. And we did structural equation modeling to confirm that, and we could then confirm that the path of causality was indeed the carbon use efficiency linkages to biomass and soil carbon accumulation. Then we looked at the data sets across the two different treatments of land use intensity. A more pristine grassland or less intensive grassland was compared to a more intensive management grassland or arable cropland, and this was not uniform because these are just sites that we found where there were contrasts that were available. So you can't really find such long-term experiments easily. So what we did was looked at the fold change on intensifications for a lot of the microbial traits that we measured, but also for some of the ecosystem properties such as soil carbon. So what we saw is on intensification, and this is only the 11 land use contrasts that are above pH 6.2, because below pH 6.2 things were very different. These are very acidic systems. So I'm not going to go into the detail for that, but there is more information about that in the paper that's published in 2018. So soil pH goes up on intensification, and this is often the case. Moisture goes down because soils lose their aggregate structure. On intensification, soil carbon is lost. Carbon use efficiency usually goes down. So we have lower carbon use efficiency in these intensive systems. Turnover rate tends to be higher, so they are growing really fast. Biomass decreases on intensification, and biomass-specific respiration tends to remain the same or increase on intensification. So this really suggests that in these intensive systems, you have a very wasteful sort of growth. Microbes are growing really fast, but their efficiency is low. So the overall growth yield will be lower, and therefore there is a lower biomass that's available to form soil organic matter. And then we tried to look at why the growth yield is lower in these intensive systems. And we did a couple of extra analyses to really understand the the details. So this is some of the proteomics work that we did in collaboration with the Helmholtz Center, with Nico Jemlich in Leipzig. And what we found here is across some of these sites, in the low intensity, which is the more pristine grassland, we saw a lot of proteins for transporters. So here resources are ample, so microbes just have to take the substrates in, whereas in the high intensity treatment, we saw a lot of stress proteins because these were stressful systems for microorganisms. They are dry. We take away all the plant matter, so there are limited substrate inputs for microorganisms, so resources are limited. So we did see a lot of stress proteins that were linked to some of these RNA degradation pathways within the KEGG classification of functional genes. We then also looked at various extracellular enzymes. We measured their activity using extracellular enzyme assays, which are mostly based on the fluorometric measurement of substrate breakdown. And what we did then was measure the carbon enzyme activity per unit biomass, and then we tried to correlate it with growth yield, measured here as community carbon use efficiency. And we found a nice correlation here with increasing enzyme investment. We found decreasing growth yield, which means that microbes are investing a lot more into extracellular enzyme production, whereas when the growth yield is high, it usually the enzyme investment tends to be lower. But then there is a lot of noise in the data as well, suggesting some extra factors. But we then also try to overlay the different sites onto this. And what we found here is that all the pristine or the low intensity sites were clustering here. So these are all the sites from the pristine habitats where carbon use efficiency or the growth yield is higher and the enzyme investment is lower. Remember here, we also found a lot of transporters because substrates are ample. So microbes are just taking in the substrate and they don't have to produce extracellular enzymes. Whereas in some of these low carbon soils, and these are intensive arable croplands, there are limited resources, limited organic matter for the microorganisms to access and take up. So what they do is they produce a lot of extracellular enzymes to mine organic matter from possibly soil organic matter, because easily available substrates are not around. So this is a nice demonstration of that trade-off that I talked about initially between the two key traits of growth yield and resource acquisition. So to summarize the work from this particular experiment, we found that in the pristine habitats, the growth yield of communities is higher. And this is because resources are ample, so they don't have to invest into extracellular enzymes. They have transporters, which are probably not that expensive, to take in the substrates that are available as root exudates or breakdown products of decomposition. Stress is lower here as well, so they don't have to invest into stress tolerance mechanisms, which was the case in the intensive croplands, where we see indicators of a lot of stress proteins in these systems, as well as a very high investment in extracellular enzyme production to mine resources from the soil organic matter. And then this has consequences for soil carbon sequestration. We saw lower soil carbon accumulation in these intensive systems because of historical losses. But perhaps one way of restoring carbon in some of these degraded soils is by providing microbes with the right microhabitat conditions and that could be just increased inputs, which will lower the resource scarcity and therefore improve growth and biosynthesis, which then also translates into improving the soil structure, because the organic matter, the necromass that's produced by microorganisms, can then interact with the mineral matrix, form aggregates, and therefore the soil becomes more um, soil becomes better, and therefore microbes will, over a period of time, see less stress and start building more and more carbon. And this is something I'm trying to study more in detail, because some of these are just outcomes of this study, which needs to be further investigated. And the second experimental evidence for the trade-based framework comes from sunny California. This was part of, again, a bigger experiment at the Loma Ridge Global Change Experiment, which is a little inland from Irvine in the Santa Ana Hills. And here we have a long-term precipitation manipulation experiment that's been going on since 2007. And some of the experiments that have been done here previously suggest that decomposition is lower under drought. So we have these big sheds, which are covered only during the winter season, which is when most of the precipitation happens in these Mediterranean systems. You can see the greater Los Angeles suburbia here and the smog around there. Regardless, it was really nice to live for a couple of years in sunny California with beautiful beaches. So why we are studying drought in Southern California? It's because future precipitation patterns through model predictions suggest that some of these Mediterranean systems in the southwest of United States will receive lower rainfall in the future. So the predictions here, the purple areas are where precipitation is likely to decrease, very similar to the Mediterranean ecosystems in the Mediterranean, around the Mediterranean Sea. So what we did here was build litter bags across these 4 treatments. So the ecosystem here is a mosaic of grass and shrub. So that really allowed us to study 2 different ecosystems and 2 different litter or vegetation types, grasses and shrubs. And we had the precipitation treatment being ambient and reduced being about 40 to 50% reduction in precipitation. And I have to really thank Claudia here, who works with Jen Martini, who helped me set and coordinate the experiment. So what we did here was place the litter bags in these cages, and they were placed on the soil surface. So grass and shrub litter separately, and then we hypothesized here that there are 2 gradients. One is of stress, which is along the precipitation gradient. So the drought treatment will reflect increased stress for microorganisms, mainly in the form of osmotic stress, but perhaps other stresses, which we will see later. And along the vegetation gradient, we saw that we saw a resource gradient. Shrub tends to be composed of more recalcitrant litter, higher amount of lignin, and lower amount of cellulose and hemicellulose and soluble organic compounds. Also higher amount of aromatic compounds in shrub litter. A lot of this shrub was coastal sage, which is very waxy as well. So what we hypothesized here is that in the grass ambient precipitation treatment, conditions will be the best for microorganisms, because here the resources are easily available, accessible, and there is less stress. So microbes will excess in the high growth. Microbes will excel in the high growth yield strategy. In In the drought environment, stress tolerance will be one of the traits that will be predominant. And in shrub communities, the resource acquisition trait will be dominant because this is a more difficult to degrade substrate, which will need some extra investment in extracellular enzymes of various kinds. And we also then hypothesized that the litter decomposition rates will be the highest in the high yield strategy. And there is no soil involved here. So we are not talking about stabilization mechanisms. We are only looking at decomposition. So a high yield strategy here would mean high decomposition rates because there is a bigger pool of biomass that's then breaking down organic matter. And then the second highest decomposition rates will be in the grass drought treatment, followed by shrub ambient, and the worst performing communities will be in the shrub reduced because here the substrate is difficult, but also there is the additional stress of drought. And we measured a whole lot of things here. So we looked at the ecophysiology of the microbial communities using metagenomics to look at the populations along these different in these different treatments and whether they have different life history strategies. We then did metagenomic assembled genomes through genome assemblies. And at time point 1, we also looked at transcriptomics and metabolomics to look at the short-term response of microorganisms to a wet up. But the results of the wet up I won't present here, but they are already published in an ESME paper that came out last year. So what we have here is a field scale precipitation manipulation. Remember, we started the experiment in August 2017, but remember that there is a 10-year legacy of drought here. So decreased precipitation for 10 years. And here I'm showing the cumulative precipitation since August 2017 when the litter experiment was set up. So the first sampling point is then 3 months later, and this was at the end of the summer drought period, so end of the dry season. The second time point is after the wet season in spring. And here you can see that there is decreased precipitation in the drought treatment because the sheds were covered. Then we have a pretty dry period through the summer, and we then sample again at the end of the dry season. And then some of the sheds are covered to simulate a drought treatment in some of the plots here. And therefore, we see the fourth time point here after the wet season. And here you can see significant amount of reduction in precipitation, nearly 50% of the ambient. And this is how the grass litter looked at the end of the experiment. And this is how the shrub litter looked at the end, almost breaking apart. And this was around, there was around 50% reduction, 40 to 50% reduction of the grass litter. So I'm showing the litter mass loss here. And in shrub, lower amount of decomposition happened. What I'm showing here is time T2 and time T3, and we sampled also an extra time point between these two time points. I'm not showing the last time point because we could not really get reliable data. But what you can see here is that vegetation has an impact on the decomposition rates. Grass has higher decomposition rates than shrub, but the precipitation impact is not very clear. Although there is a trend in the direction of higher decomposition rates in the ambient grass treatment, but this wasn't significant. But I just want to highlight that previously we've seen significant differences in decomposition rates in the grass ecosystem, in the grass vegetation. So it really depends on the time point of sampling and the seasonal fluctuations. But nevertheless, we saw higher decomposition rates in grass than in shrubs. And I'll come to this towards the end after I explain some of the ecophysiological patterns in the data that was generated. So the microbial community data from metagenomics allowed us to look at how the communities progressed during decomposition. So looking at community succession during decomposition. And here I'm showing some ordinations, taxonomic and functional classification. I'm gonna just pause and close the door, sorry. Sorry about that. There was just a lot of noise. So what we saw here is that during community succession, we have the taxonomic communities, the taxonomy of the different communities merging together at the end. And we see that the shrub communities are very different. So squares are the shrub communities, and these are the taxonomic profiles. Triangles are the grass communities. So they are different. And within them, you have the drought treatment, which is also significantly different, at least initially, if not later. But you can see that as decomposition progresses, there is convergence in terms of the taxonomic profiles, but also of the functional profiles. And actually the functional conversion is higher than the taxonomic conversion. So this just shows that the decomposition, as decomposition progresses, the communities become more homogeneous and the litter probably becomes more uniform, including microbial biomass becoming a source of substrates for other microorganisms. In terms of diversity, taxonomic diversity of the communities, it increases as decomposition progresses in both grass and shrub communities. So these 8 points here are grass communities, the later 8 points are shrub communities. Blue is ambient precipitation and red is reduced precipitation. And the 4 points are time point 1, 2, 3, and 4. So the progression of decomposition. And what I'm showing here is that as decomposition progresses, the diversity increases. And this is mostly because initially there was a very high dominance of fungi in the leaf litter communities. And as the decomposition progressed, bacteria started to take over, and there were just a very diverse range of bacteria that were then dominating towards the end, which is why you see increased diversity towards the end of the decomposition process. So we see that the fungal bacterial ratios are higher initially, but they're also higher in the grass communities compared to the shrub communities. And there was not a significant difference across the precipitation treatments. Now, to dig into the functional changes during decomposition. So this is these are all the level 1 functional categories of all the functional genes that we could annotate through our metagenomics data sets. And what I'm showing here is the fold change in the abundance of these functional genes within these categories at the broader scale to drought. So what you see is in the red, an increase in the change with drought. So higher abundance of these functional genes in the drought treatment compared to the ambient treatment. And what I'd like to highlight here is 3 main functional categories of microbial functions. which tend to dominate in the drought ecosystems. And these were linked to membrane transport, stress response, and ion acquisition and metabolism. And then we went deeper into looking at how these three main classes, functional categories, change with time. So the membrane transport, again, you can see higher amounts of reads that were annotated to membrane transport in the drought treatment, both in the grass and the shrub treatment. Higher amount of genes annotated to stress response functional categories, both in grass and shrub, although in shrub it wasn't significant. And ion acquisition and metabolism as well, significant at many of the time points, both in the grass and the shrub vegetation. So these are the three main functional sort of indicators of drought. So microbes producing stress response, and a lot of this was very much linked to osmolite production or osmolite synthesis, so that the microbes could maintain osmolarity in this new drier environment. So what we did here was genome assemblies, and then the scaffolds were then annotated using COFAM, and this is data before binning. So we are looking at the gene domain numbers, or the diversity of genes that we could find in the metagenomics data sets. And using DSeq, we looked at the most abundant genes that were found in the three main groups that we identified, the three main functional categories that we identified, that of membrane transport, stress response, and ion acquisition and metabolism. And within these two were linked to maintenance of osmolarity using inorganic or organic ways. So glycine betaine, for example, is a very classical osmolite that's used not just by microorganisms, but also by plants to overcome osmotic stress. And you can see that the number of genes are higher. So these are genes that are present in unique populations. So there are more populations having those genes in the drought treatment in both grass and shrub vegetation. And I'd like to acknowledge work by Antonio Ribeiro, who's working he's at the Genome Center in Aberdeen, and he's working part-time with me through the funding that I received to start as a starter at Aberdeen. Then we wanted to also look at the resource acquisition trait. We looked at the stress tolerance mechanisms, which are mostly linked to osmolites or maintenance of osmolarity. In terms of resource acquisition traits, because most of the substrates that were present in the leaf litter were linked to different carbohydrates, we looked at the carbohydrate functional category in the subsystems classification from the metagenomics dataset. And we also measured a suite of carbohydrate enzymes, the activity using the fluorometric extracellular enzyme assay. And both these datasets quite mirror each other. We see that in the grass communities, there is higher carbohydrate genes. There's also higher carbohydrate enzyme activity. Then in the shrub communities, the precipitation treatment is not very clear, although there is some indication that in the grass ambient, there is higher abundance of carbohydrate genes than in the drought treatment. But overall, we did not see any evidence of constraints on decomposition machinery in the drought communities. And this was again reflected by some of the population level data set that we looked at. So these are genomic assembly scaffolds before binning again, which were then linked to Cazyme, not COFAM, Cazyme database. And again, here I'm showing you for grass and shrub, ambient and reduced, all these different categories of Cazymes, GH genes being the most abundant. And what we see here is that except for maybe one of the first time point, generally the number of unique genes that we could found in unique populations in these two different ecosystems was not that different. It was not statistically different. So again, resource acquisition doesn't seem to be impacting that much across the precipitation treatments, but we can see differences across grass and shrub, obviously, because the substrates are very different. And then finally, I just want to show the yield trait. So these were measurements which were a bit more quantitative, not from the metagenomics data sets, but we measured biomass using DNA concentrations, and we measured respiration using infrared respiration infrared analyzer. And what we see here again is that there is no treatment, no significant difference across the precipitation treatments, but we see that in the shrub communities, the biomass is lower, and in shrub communities, the QCO2 or the biomass-specific respiration is higher, which again signals to the fact that there is higher physiological constraint in these shrub communities because the substrate is more complex, which is why they are growing slower, the biomass is lower, and they are respiring a lot more per unit biomass because there is a lot more of maintenance respiration happening because the substrates are a bit more difficult to break down. So again, across the precipitation treatments, we did not see evidence of growth constraints. And this could mean that the microbial drought adaptations that we saw through the various stress tolerance mechanisms, mostly linked to osmolyte, are probably not giving the microbial communities, the populations within the communities, an increased amount of fitness so that their decomposition capabilities and growth is not really getting affected by drought, because some of this investment is likely not that great, and the adaptation that they've formed is allowing them to grow really well or grow at the same level as the ambient treatment. And finally, I just want to show why that's the case. And this is some of the litter chemistry data, which will allude to some of the mechanisms that I just showed. We did here FTIR measurements to look at the litter chemistry. This work was done at the Berkeley lab. And again, thanks to Shi and Zhao for the excellent collaboration on this front. What we saw here is if we do a quick ordination, the shrub and grass litter is very different in terms of the different functional groups of chemical compounds that were found. And we see that there is a precipitation treatment as well. And this is then showing quite different leaf litter in the grass ambient and reduced, but also shrub ambient and reduced, although in the shrub litter, the differences were smaller. And that then again is a nice reflection of the carbohydrate genes data set, which again shows that there is a very small difference in the shrub litter, but somewhat of a difference in the precipitation treatment in the grass litter and the grass communities. And in terms of what compounds we found in the grass litter, we see that there is higher amount of glycosidic bond carbohydrate compounds in the ambient precipitation, which is lower in the drought treatment. Same for some of the other carbohydrate compounds, although it's not very significant. But in drought treatment, the litter had higher amounts of proteins and higher amounts of lipids. whereas in the shrub, the differences were not that high. But if you compare grass with shrub, you see that there is lower amount of these easy substrates, but higher amounts of lipids and higher amounts of CO stretching carbohydrates, which probably explains some of the lower rates of decomposition in the leaf litter. But what you see here is that the drought impact, especially in the grass litter, is mostly because of the changes in the above ground leaf litter, or the changes in the plant communities, and hence the plant leaf litter, and less of an impact of direct drought stress in terms of changes in the moisture. So with that, I'd like to just summarize the second data set that I presented. Here we hypothesized that in the shrub litter, decomposition will be higher, and that was the case. Sorry, grass litter decomposition would be higher, and that was the case. It was lower, slightly lower, although not statistically significant in the drought treatment. And we couldn't really see the drought treatment making a big change in the shrub communities. It was very similar across both of those, although shrub communities overall had a lower decomposition rates because the litter there was more complex, and we also showed how the different cazymes and different carbohydrate genes and their activities, the extracellular enzymes was also lower in the shrub communities, and therefore the decomposition rates were lower. So we nicely linked this YAS trait structure to the decomposition rates that we could measure, and then that allowed us to explain some of the mechanisms behind some of these processes. So with that, I'd just like to highlight some of the work we now do in University of Aberdeen at the newly established Microbial Biogeochemistry Lab. We continue to look at microbial traits, or looking at the ecophysiology of microbes with a particular focus on soil carbon cycling. But we are trying to do more of a population and community scale work, again, trying to integrate that to ecosystem ecology so that we can answer some of the key societal challenges of sustainable land use, climate change resilience, and peatland restoration. Peatland restoration being one of the key national challenges in Scotland. So with that, I'd like to thank you again for your attention, and all my collaborators. And thank you to John and Karen for the invitation to give a talk here. I'm happy to take questions.
Ashish, thank you so much. And thank you so much for enduring the unwanted visitors on that call. Thank you. And huge apologies from both me and John for any issues and any offense that anyone may have taken from the content. Fantastic talk as well. Thank you. If anyone has any questions, I know we're over the hour now, but I'm very happy to stay. I don't know if you can as well, Ashish, and John as well.
Yeah, I'm fine.
Okay, so
I love the background noise, Ashish. Actually, the kids playing in the background. It's part of our Zoom life these days.
Yeah, completely. Does anyone have any questions they'd like to start with? Hongwei.
Hello, I have, thank you for the nice talk. It's very interesting. And I have a question regarding to the community carbon use efficiency. I imagine the plant communities will influence the microbial community, which will determine the carbon use efficiency. And on top of that, which material feed to the community will also influence that. So I was wondering which material, how do you decide which litter you feed the community to get the CUE?
Oh, so we used a generic shrub litter, and we grew it. We dried the leaf litter from from a temperate shrub called Chenopodium, and it was ground, and then we created a solution and then filtered it. So it was a DOC that was labeled with 13C when the plants were grown. So it's a easy substrate, or relatively easier substrate for microorganisms. And we just used that because it was a generic substrate. We didn't want to change it. Obviously, it will affect the amount of CUE, but we used the same substrate for all our treatments, if that answers your question.
Yeah, I was wondering if you have chosen another substrate, do you expect the result changes?
Probably not, but what we used DOC because we didn't really want to use glucose or some of the easy substrates, because that may really affect the results. Because glucose tends to be, would be metabolized very easily by almost all the communities, and perhaps not really give us those differences that we would expect across those treatments. So just trying to simulate the natural environment as much as possible.
Thank you.
Any other questions from the audience? I have a quick question, whether you did much analysis into how much, for example, the first grasses experiments, how much that translates into the nutritional value of those grasses. Were you also looking at that, or do you stick only with the soil side of things?
Yeah. Do you mean from the California experiment?
No, the first one, with the pristine grasses versus the over farmed.
Yeah. No, we didn't really look much at the plant level, but many of these were comparisons of just meadows, which were either abandoned or just there was a light mowing, and comparing that with just cropland. So it was very different plant communities. Sometimes the differences were smaller in the sense that we had just small difference in intensification across the treatments. So it was still farmed, but one with a more intensive, with deep tillage and all of that, and the other one was a more sustainable way of farming.
Yeah, because there's quite a lot of I certainly hear a lot from people trying to discuss this idea of less over farming of landscapes. So it'd be really interesting to see your work on that, and then also tie it into what they want out, which is good plant material for their fodder or whatever it is.
Yeah.
Rosa, did you have a question?
Yeah, thank you first of all for this brilliant talk. I really enjoyed that. I had a curiosity. It's probably a little unrelated, but I had the sense that you may know something about it. Do you know if there is any data around on the effect of plastics on plastic pollutions, of course, on microbial communities in the soil?
Not really, but there is a lot of research going on. I know Matthias Rieling's lab in Berlin is doing a lot of work on microplastics in agricultural systems. Yeah, but sorry, not
no,
really.
No, that's fine. Actually, can I email you to ask you about his name again? Just
yeah, sure.
Thank you. I'm trying to understand if the community who knows something about that.
Yeah.
Thank you.
Happy to help.
I have a question, if I might. It refers to something that you alluded to in your talk, but you didn't focus on it. I think you referred us to an ISME paper. I wondered if you could just give us a quick snapshot, though, of what you learned in the wetting experiment situation, the wet up situation. I'm curious to know if you saw lags between, or any cyclicity across the sort of wetting up, drying down experiment?
So we looked at transcripts, so changes in gene expression to a local wet up, and we also did metabolomics to look at some of these osmolytes as well as some of the biogenic amino acids and how they change with the treatment. So we wet the litter and sampled the day after, and we also sampled 12 days after, and this was a dry period. So it let us look at the wet up and dry down. The first thing was the experiment itself wasn't successful because there were local effects. We did wet up, but after at the day one sampling, some were already quite dry because they were exposed to the sunlight, whereas, you know, a replicate which was shaded had more water. So the differences were not massive. And because it's such a short pulse that you get of respiration and increased growth, it wasn't, so the legacy of the drought wasn't having much of an impact on the wet up response. Also good to highlight here at this stage that California has summer drought. So the microbes do see drought during various stages of their lives, you know, like they are adapted to that. So it's not a new thing to them, which is maybe one of the reasons why we are not seeing big changes, big impacts of drought on just growth or respiration.
Yeah.
But some of the indicators we saw were mostly linked to osmolytes. When you wet up, you have some of these osmolytes for glycine betaine or some of the amino acids which are used as compatible osmolytes. And as soon as the drought goes away, they burn it up. So it went down with wet up and it went up again after the dry down.
Yeah, that makes sense. Yeah.
And there were some genes for flagella which were increasing in expression. And in terms of what was going down also was some of the stress proteins. So chaperones, which were linked to, again, maintenance of maintenance of integrity of biomolecules.
Yeah, thanks. I really, by the way, I really like how your work just covers, you know, metagenomics, transcriptomics. You've used FTIR. It's a really great spectrum of molecular and spectroscopic approaches. I think there's another question in the audience.
Yeah, go ahead, Hongmei.
Hongmei, is that a new hand or your old? That's a new hand.
Go ahead. I was wondering, in the second experiment, what's the contribution of solar radiation to the decomposition? Because you just place the little bags on the surface of the ground.
Yeah, so it is big, and there are some previous studies which have shown photodegradation. It tends to be quite significant if it's like exposed litter bags. And like I said, some of the litter bags were more shaded than others, but overall they were placed under the canopy. It affected the one-day experiment because it was, you know, like collected during noon. So probably the shading was on a particular side more than the other. But I think overall it didn't affect our experiment, but there are some other experiments where they've shown the contribution of decomposition rates from chemical processes.
Yeah, especially when you have much more lignin in the litter than it. And just a quick question. Did you measure the microbial community related parameters from the litter? So did you get to?
Yeah, we've previously done that, but this was litter collected from the same field, which was put back in there without any treatment. So we hope that it's the same community without any changes.
My question was, you measured a lot of microbial community related parameters. Did you get the microbial community from the litter you put in the bags?
Yeah, so the litter bags were quite open. So we think that we take the litter from the same environment and we put it back in the same environment. And after 3 months, we do the first analysis. So we think that there's enough dispersal from around the leaflet, from the litter bag, that eventually it sort of, becomes the same as the litter outside of those cages. Do you know? Do you get
Maybe
what
I
I mean?
didn't put my question clear.
Maybe that's something you could follow up with Ashish
Yeah.
afterwards.
Yeah.
if that's
Okay, sure.
Before he fades into the dark here for
I can put my lights on.
No, we thank you, everyone. We obviously had some difficulties starting up, so we let ourselves run over a bit. But should probably thank the speaker one more time for a great talk. Thank you, Ashish.
Yeah, amazing, Ashish. Thank you so much.
You battled through a lot of noise and distress.
And so we're back again in 2 weeks' time with Charles Cockle talking. I forget the title. Sorry, something along the lines of the
Geobiology
Z-axis.
in the Z-axis.
Yes, that's the one.
Fascinating.
So I hope you all join then.
From what I understand, that's positive Z and negative Z as well. So we're going up and into the subsurface too.
Mind-boggling.
Yeah.
I'm looking forward to some of the talks. And thank you again for having me.
Yeah.